This webinar is about 2D gel experiments, project setup with Delta2D. Let's first talk about project goals. Typically, when you want to start a 2D gel experiment, you want to answer a biological question with it. And you want to publish this answer in a paper or on a poster at a conference. Furthermore, you have to meet some restrictions. For example, you have to finish your project within a certain timetable and also within a given budget, because grants are just given in a limited size to your project. Before starting, you should check yourself. Ask you the following questions. Am I able to produce good gels repeatedly? That means, do I have standard operating procedures so that the technical variance within my electrophoresis steps is low enough? Did I define good milestones? That means, can I measure intermediate results that I can analyze and interpret to make decisions with respect to the progress of this project? Furthermore, will I keep records? Will I document all the parameters that I have defined, the decision that I have made, and the consumables that I have used? This is important because you want to be able to answer all the questions that come from reviewers, from colleagues, or from supervisors. The most important question maybe is, am I prepared to stop my project? It could happen that the intermediate results at a milestone look that poor that you'd better start again, at least if the amount of sample is sufficient, rather than to continue the project, waste more time and more money with it, and then getting poor results that are not worth to be published. The planning of your experiment should be very careful. Ask colleagues, ask other scientists that work on the same organism or maybe with the same technology, how they designed their experiments. Look into papers, how others did this work. Furthermore, randomize as much as you can. If you don't have enough samples so that randomization would work, simulate randomization. That means distribute your samples equally across the different working days, technical assistants, or maybe also buffer batches or gel runs. Furthermore, we recommend that you use more replicates rather than more factors. If you add more and more factors to your project that you want to analyze, then you run into the problem that the project becomes as complex that you do not have an overview anymore. You get lost in finding answers to your questions. For different questions, for different factor analysis, prefer to analyze different projects and experiments. Furthermore, prefer biological replicates instead of technical ones. Biological replicates also always include the technical variance. Furthermore, if you use technical replicates, do that to determine the technical variance. Consider pilot experiments for doing this. In a pilot experiment, you could just run technical replicates. With this way, you measure the technical variance and then know it, and hopefully it's stable enough since standard operating procedures are used. And when interpreting real results later on, then you would be able to distinguish between that part of the variance that is due to technical reasons from that one that must then be due to biological reasons. Let me stress again, use replicates. Always work with replicates. Consider a protein A, where we theoretically could observe 15 data points of a control and 15 data points in a treatment sample. However, nobody is able to really use 15 replicates. You typically have just 2 or 3, and if you just get randomly selected data points, that's not a process that you really can control which data points you will get. You might only see these 4 data points, and since they have the same level here, the same protein amount, you might interpret that as no change between control and treatment. This would be a false negative finding, because if you would get the green highlighted ones, then you would interpret that as a change, would follow up with some statistical analysis and would find that yes, there is really a change, not only in the mean but maybe also in the p-value between control and treatment. So this finding would be okay then. Consider protein B. Again, 15 data points for control and treatment theoretically available. You just get 2 of each, and then these 4 highlighted ones would mislead you to a false positive finding. You would think there is a change between control treatment, while obviously there is none. On the other hand, getting these green highlighted data points, you would then say, no, there's no difference between control and treatment, and this conclusion would be okay, since the average between control and treatment is just the same. So that means we can have irrelevant proteins, there's just random change, no real biological finding, and there are proteins like protein A with systematic changes because of biological reasons, and there is a discovery inside. What could you do? What error can happen? You could miss this kind of discovery and not publish it, not validate this protein. This is less likely with bigger effects between, for example, control and treatment samples, or it is less likely if you run more and more replicates. However, if you misinterpret an irrelevant protein to be relevant, if you misinterpret a random change to be a change, then you publish a false positive, and this has been based on wasted time and also money in validation of this protein. So you waste resources, publish a false positive, and that indeed shouldn't happen. The p-value of a certain expression profile of a change between a group control and a group treatment is giving you the probability of making this error, and this error can be reduced, the p-value can be reduced by the number of replicates, provided that the technical variance is under control and is low enough. Let me now give you an example of randomization, for example for dye swapping. Consider a dye experiment. In dye experiments you can run multiple samples within the same gel, since all these different samples are labeled with different fluorescent dyes. One of the dyes is used for the internal standard to increase the reliability of your quantitation. There are some aspects with that. The excitation wavelengths and also the emission wavelengths of the different fluorescent dyes are different. That means you need imaging devices which can handle these fluorescent dyes. You need light sources, lasers, for example, or LEDs that just excite at wavelengths that really excite the correct fluorescent dyes. And you need a sensor or filters that just measure the wavelengths emitted by a respective fluorescent dye. However, there can happen some things, for example crosstalk, also known as bleed-through, can happen when the emission wavelengths are not that well separated for two different dyes, but they would overlap. If you would then like to measure the complete red signal, then you would necessarily also measure part of the blue one, and this would lead also to misinterpretation, mismeasurement. You would measure a quantity which is not true. For this reason you should apply then filters which only filter out this part of a certain emission spectrum which does not contain emission from other fluorescent dyes. However, the most important aspect is labeling efficiency with respect to dye swapping. Labeling efficiency means different dyes might bind with different efficiencies to proteins, to different proteins, because of maybe the 3D structure of the dye or of the protein. And also the emission intensity could be different between the dyes. And that means you would get a systematical bias between dyes for the same proteins or for different proteins. And this bias should be randomized and averaged by dye swapping. So then you do not label a certain sample and all the replicates of this sample with the same dye but with the different dyes. With this you average the effect of this bias and do not measure a wrong quantity. Let me give you an example. For a diet project, consider a cell line where we want to analyze two factors. One is treatments, so we split the cell line into 4 cell lines. One is the control, which remains untreated, and 3 others are treated with 3 different treatments. We want to observe these cell lines at 3 points in time, and then we want to take 4 replicates for each point in time. That means we will get 4 times 3 times 4, a total of 48 samples. On dyed gels, up to now, you can run 3 different dyes. One is taken for the internal standard, so 2 are remaining for the samples. That means we need 24 dyed gels for 48 samples. How shall we assign the samples to gels? Consider the control sample and the 3 points in time with 4 replicates each. Then we start with gel 1 and continue with the replicates until gel 12. That means we distribute all the replicates across 12 different gels. That means we do not only swap the dyes but also the gels. Regarding the dyes, we have the G200 in Sci 3 and G300 in Sci 5. They are used in an alternating way for the replicates. This results then in 12 different images. Having numbered the gels like this doesn't mean that they would have to be run in the same order. Of course, we need to randomize the gels across the different gel runs as well. Continuing with treatment 1, that means we start then with gel 13 for the first replicate, continuing then to gel 24 for the last replicate of the treatment 1 sample. The dyes are then used or applied in the same way. Beginning with treatment 2 then, we start again with gel 1, assigning it to the first replicate of this treatment, and then since G200 on gel 1 is already used by the control sample, we need to take G300 or the Sci 5 dye and continue then with G200 Sci 3, which is the free dye for gel 2 here in this example. We continue with that also for treatment 3, and then we have used the G200 and G300 dyes on gels 1 to 24. The G100 or Sci 2 channel is always used for the standard sample, which is the average sample across all our samples. This results then in 24 more gels or gel images, number 49 to 72. So a total of 72 images will result out of this experiment. However, imaging is not that easy as it looks like. We have often seen that images have been looking rather poor, but the gels behind seemed to be fine. So something might have happened during the imaging process, and that's why we have written the imaging guide, which is available on our website as PDF, where you find essential tips and tricks for imaging. It includes a lot of information about dyes and also imaging devices like camera systems or scanners, and you can avoid problems like choosing a wrong image file format, a wrong resolution, or how you should optimize the photomultiplier voltage or the excitation or the exposure time on a camera system. Having imaged your gels, then you should be careful with post-imaging image processing steps. If you want to rotate your images because maybe the gels have been scanned with the wrong orientation on the scanner or in the camera, then you can rotate them, but only by 90-degree steps. You can also mirror them vertically, horizontally, that doesn't matter, and invert black and white. All these steps wouldn't change a pixel. Everything should be reverted or could be reverted if necessary. But we also recommend to use one pixel manipulating image processing method, and we recommend to apply a speckle filter if necessary, since speckles disturb the analysis process very much. So it's much better to apply a speckle filter, even if it is not changing the remaining spots, rather than to analyze images with a lot of speckles on them. A speckle filter is, by the way, included in Delta2D. How does this project now look like in Delta2D? We have the cell lines, a control and 3 treatment cell lines. We have 3 points in times, 0 minutes, 30 minutes and 60 minutes, and for each such combination of a cell line and a certain point in time we have a group containing 4 replicates. What we cannot see here are the standard images. They look all the same and are labeled with the G100 die, and they are not present here since they do not contain real sample information. This project, by the way, has been created by Christian Scharf. Thanks to him to helping us with this project so that we could create this nice view and can use it for other training purposes as well. Now let's have a look where the images come from. The first image in the 0 minutes group for control and treatment 2 come from GIL 1, while for example the third images in the 30 minutes group for treatment 1 and treatment 3 come from GIL 19. We can also have a look how the images are labeled. You can see the green highlighted images labeled with G200, while all the others are labeled with G300. Let me now show this in Delta2D. I start Delta2D, and then, having started it, Delta2D will offer a list of demo projects where I can choose to open one of them. Is a demo project for a regular element, which is the demonstration project. You can see it in this list as the first item. The other one is a Deutsch project, an example project. Raw data is available on our website. You can at any time just open them, surf in, change them and play with them. Here is the pool path provided. That means this is the path to the folder where this project or these two projects are saved in. However, I don't want to mix my new information, my new experiment with these demo projects, because that's also what we recommend. Save your own experiments on a different location on your hard disk, maybe where you save all the other experimental information as well. That's why I click Change Pool. Now I can surf my file system, go to my webinar folder, and then create a new folder there, which I call webinar pool. This is a regular folder, and pressing OK means Delta2D will now create a structure of subfolders and files which makes this folder a Delta2D pool. This pool is now empty, there's no project inside. That's why I click New, enter a name for a project like this. Enter my initials as the author, and then I click also Use Internal Standard and add a description like example project for webinar. And then I press OK, and then I can open this project. Then, at the beginning, Delta2D is always creating two groups. One is for maybe a control, the other one for a treatment, and that's the minimum number of groups that makes sense. We always want to compare something. In the left, we have the workflow panel. The first step is already opened. We have some detail information, the ones that I have entered, my initials, the description of the project. I could also now open a dialog to import images to my pool without importing them to the project. Here's a list of groups. I can add images to each group or add a new group. And I would like to add images now to group 1. I press this add link and I get a dialog with a list of images in my pool, which is empty since this project and pool are completely new. I press import images. Again, I get access to my file system. I choose the webinar folder, and here we have folders for 4 gels. I click the gel 1 and see that there are 3 images inside for the 3 different dyes. Then I press next, and in this property dialog you can see that the gel image name as it is represented in Delta2D is automatically taken from the file name. There has been detected that the image has been imaged with a typhoon and a calibration has been used, and also that in the file name the G100 string shows that this image is labeled with G100. In this area we have this the simple operations that I have mentioned. We can mirror an image horizontally or vertically or rotate it by 90-degree steps. Doing two mirroring steps and two rotating steps means we end up with the original orientation here. And furthermore, there is a zoom preview. I can move this rectangle to an interesting area, and there we can see how this part of the image is looking like. And this is important to control whether there are speckles in the image. If there would be speckles, I could apply a certain filter for black ones or for white speckles. Since there is no speckle, I don't have to apply a filter here. I can also invert the image and have the review how it would look like here in this preview. Inverting is necessary if images come with white spots on black background. This can happen, and we have to invert that then to black spots on white background, since Delta2D algorithms are based on this setting. Then I press next, and that get the same dialog for the second image that I have selected. And since I know that the gel1G200 contains a control replicate, I can just assign this to the control sample in this dialog. Everything else is fine, and I can press next for the third image, and everything is fine here as well. I press finish now to import these three images into my first group. Here we go. And as you can see, the three images are in the same group, but groups should always contain replicates. First, I remove the standard image from this group. I just drop it on an empty area on my an empty space on my area here, and then there are just two images remaining. And since I have used die swapping, I am sure that this gel1G300 is not the control, but is containing the treatment. By the way, the small example that I will show here contains just one factor. I will just distinguish between a control and one treatment. Otherwise it would be too complex to show it in this webinar. As you now can see, we have three groups. One contains the standard image. That's why I would like to change the group's name. I double click its name and enter standard. Group 2 contains the treatment. I double click the group name again and call the group treatment. And the first group contains my control image, and that's why I rename this group to control. You can also rename images by just double clicking their name and editing it. Let me add a second gel. I import images again from my file system, go to my webinar folder and select the gel2 images. I press next, and then I can see this is just the same as before, and then I can just click finish to import all the three images at once. Let me again move the standard image to the standard group, and then because of die swapping I know that the G200 on gel2 contains the treatment image. That's why I move this image to the treatment group. Let me now add more images. This list is still empty because the pool isn't empty. There are images now, but they are all already assigned to my project and there are no images left to design here. That's why I need to import further images from my file system. Let me continue with gel3, and I can just press finish to import them as well. I have clicked the add button here next to the treatment group. That's why they show up here. Let me move the standard image to the standard group again, and then I have to be careful again. gel3G200 is again control, and this way I would also like to add the fourth gel. like this. Select all these images and press finish, and then they are all imported again. I move again these images to the respective groups. Well, that's what Delta2D needs to know to analyze this as a regular experiment. However, this is a Deutsch experiment and we have to let Delta2D know which images come from the same gel. Delta2D could take it from the name, for example, or from the fact that they come from the same folder, but this is not always necessarily true that these are indicators for the same gel. That's why we have to assign that manually. Here's access to a gel image attributes dialog, or I can just click this gel image attributes link in my workflow panel, and here we have a list of our images and also some predefined gel names. I would prefer that they are not numbered with these Roman numbers, but that's why I right click on the first, choose properties and rename this one to gel1. I'll do the same with the second and with two others. Like this. And then I can just select the three images for gel1 and drop them on gel1, for gel2 drop them on gel2, and so on for gel3 and also for gel4. You can also now see that there are some radio buttons. This radio button indicates that the G100 image is the standard image within this gel, because Delta2D also needs to know which image contains the standard image to use this as the basis for normalization then. Let's have a look at this sample tab here. We can also assign our images to samples. This is not necessary for the analysis, but for reporting the results then. That's why I would like to do that. First, let me rename one of these predefined samples A to H to standard and another one to treatment. We can also rename this to control. So then I can just take the standard images, drop them on the standard sample, and then I choose gel2 G300, gel3 G200 and gel4 G300, move them to the control since they contain these replicates, and the remaining images are all treatment replicates. And then you can see for the treatment sample, okay, the dye swap has been applied, the gel swap as well, and the same applies for the control group. That's what we needed to see. Then I press OK and save my project, since this project is now well defined and the first step, setting up the project, is done. Now we could stop this webinar. However, let me just show a report which is documenting this step. It's the project summary report. We can access this via the reports menu item and project summary here, or via the project summary icon in the symbol bar, or we just open the fifth step of our workflow and click the project summary link. This is opening my web browser. It is opened in full screen mode now, and here we see some information, a header with information which describe our project, for example the description, the author, and also the path to the pool. There's a save button here. If you want to save this project summary report as HTML file, use this button. Don't use your browser's save file as function, because this wouldn't help to save the complete report. And then there's also information about the groups, a list of images for each group, their dimensions, and also the used calibration. This is repeated for all the groups. And then we have an overview in this area about the samples. There is a control sample containing certain images. They have been labeled with certain dyes and run on different gels. So this is then documenting your gel swapping and dye swapping. There's a section for warping. If you would have done the warping step already, you would get a list of the direct warp relations here in this area. Furthermore, I can click here and request images. Delta2D knows delivering the images for the different groups. This takes a second, and now they are popping up. As you can see here, here are 4 images for the control group. Then we have 4 images for the treatment group, and also 4 images for the standard group. If you would have done the warping again, we would also get dual channel images for the direct warping relations. Furthermore, let me stress that when importing the images, automatically histogram settings have been applied, the contrast has been increased so that you can easily see low abundance spots on your images. This is just for visualization, but does not affect the quantitation in any way.